The first time the idea of activating or suppressing the immune system to attack cancer was in the mid-1950s, when the only cells that could be genetically modified were single-cell organisms. Today, immunotherapy is a reality, and it involves adoptive cell therapy, using the body's own T cells that are slightly modified to fight cancer. Our body has two primary cells in our adaptive immune system, T cells and B cells. B cells excrete antibodies specific to a foreign substance, the pathogen, that they are exposed to, neutralizing them or tagging them so T cells or other immune cells can handle them. T cells destroy the pathogen immediately and are therefore sometimes known as killer T cells, or the workhorses of the immune system because of their immediate response to cells infected by pathogens. They also sometimes secrete cytokines, proteins that are involved in cell signaling. Many people with initially healthy immune systems develop cancer, a sign that the immune system cannot fight cancer on its own. This is because the immune system sees the cancer cells as the body's own cells and not foreign cells, so it is not able to locate and attack them or doesn't produce a strong enough response to eliminate them. In some cases, the cancer cells even secrete certain chemicals to disguise themselves as body cells. Therefore, immunotherapy methods are adopted to assist the T cells in recognizing and eliminating cancer cells. One of the most effective forms of immunotherapy targeted at cancer cells is known as chimeric antigen receptor T cell therapy, or CAR T cell therapy, involved in modifying patients' T cells. This therapy is currently extremely effective for patients with advanced melanoma and acute lymphoblastic leukemia. This therapy typically happens in 3 steps. The first step is collection. The patient's T cells are removed and isolated in a process known as leukapheresis and then frozen. Next, the cells are reprogrammed to express the chimeric antigen receptors, making them CAR T cells. This is done using the lentiviral gene delivery method, exposing the isolated T cells to a disarmed, modified lentivirus which contains genes to code for the CAR protein. These genes are inserted into the T cell's genome to produce T cells that express the synthetic CAR receptors, i.e., CAR T cells. The T cells are modified to become more cancer-specific. The lentiviral gene delivery method results in an artificial transmembrane protein. The CAR protein that is added to the T cell essentially consists of the antibody that is specific to the antigen that the therapy is trying to attack, and a receptor protein. This means that when a CAR T cell comes into contact with a cancer cell, it should be able to recognize the antigen on its surface and become activated, leading an attack on it, which may be assisted by other T cells. Before infusion of these cells, the patients are treated with a course of chemotherapy. The reprogrammed cells are multiplied in the laboratory for 7 days and then are infused intravenously. The reason for this course of chemotherapy is to deplete the non-modified lymphocytes before infusion to improve the efficacy of the therapy. After this, if all goes as expected, the CAR T cells will multiply within the patient's blood and recognize and kill the tumor cells in the patient's body. However, there are many possible side effects to be aware of. One is T cell exhaustion, which is when T cells stop being responsive due to changes in gene expression. This could be prevented by treating the patient with certain immune checkpoint inhibitors. Another is antigen escape, which is when some of the cancer cells do not even produce the targeted antigen required for the CAR T cells to recognize, and hence escape T cell activation and continue to multiply. This can be combated by introducing more than one kind of specific CAR to a cell during genetic reprogramming, especially in the case of acute lymphoblastic leukemia. This increases the chance of targeting all tumor cells. The solutions for both these side effects are still undergoing clinical trials. Another major side effect is the result of the overproduction of cytokines by the CAR T cells. This can result in high fever and very low blood pressure. This is known as the cytokine release syndrome, or CRS. While the side effect is dangerous, it is also a sign that the CAR T cell therapy is successfully administering itself in the patient's body, as it means that the T cells are multiplying and activating. Since this is a known side effect, doctors watch for it and treat for it early so the therapy can be most effective. Lastly, a significant side effect of CAR T-cell therapy is B-cell aplasia, a mass die-off of B cells. Some antigens present on cancer cells can also be present in small quantities on regular B cells, which produce antibodies to fight off pathogens, and as a result may be targeted by CAR T cells. Patients receive immunoglobulin therapy to produce the necessary lacking antibodies because of B-cell aplasia. In terms of applications of this therapy, as of now, CAR T-cell therapy is used for liquid tumors. However, because it is more difficult to find unique surface markers that distinguish solid tumors from healthy tissue, and because the matrix that surrounds solid tumors is immunosuppressive, it is more difficult to apply this to them. However, studies in mice suggest this may be an effective treatment as a part of a combination strategy. That being said, the possible applications for this therapy are immense. Since each immune receptor is antigen-specific and each cancer has a different kind of antigen, CARs modified in the lab can be made for that specific cancer's antigen. For instance, leukemia has antigens down as CD19, and CARs made for CD19 will not work for any other type of cancer. This means that the variations of CAR T-cell therapy can treat multiple types of cancers and possibly other diseases using a similar method. Immunotherapy is becoming the future of cancer treatment. CAR T-cell therapy has led the way for treatment of acute lymphoblastic leukemia, with the remission rates as high as 90% in clinical trials, and with combination therapy and possible innovative solutions to the administration for solid tumor treatments, it could pave the way for a more effective way to treat cancer.